A-Level Biology 9700, Paper 4, Variant 1, May June 2025. Today we are going to discuss the question number 6. And question number 6 is, in the kidney, various molecules move between the blood and the kidney nephron. Table 6.1 describes the movement of molecules at three regions of the nephron P, Q, and R. So the regions at P is amino acid. Molecules are amino acids, glucose, and water. So the direction of movement is from nephron to blood. And in Q, water from nephron to blood in the presence of antidiuretic hormone. Water and the molecules with a molecular mass of less than 68,000 from blood to the nephron. So at P, the first of all, I would like to P, you can see from nephron to the blood. It means the selective reabsorption is occurring over here. They are needed by the body. So they are selectively reabsorbed in this region. So the for P, the process which is taking place, because the direction of movement is from nephron towards the water. So they are selectively reabsorbing. Selective reabsorption. And second, you can see the water from nephron to blood. From nephron to blood in the presence of antidiuretic hormones. Again, the water is reabsorbing. So water reabsorption takes place to keep the osmoregulation in the body. So we will write osmoregulation. And this is also called as facultative water absorption. So this process in the nephron actively regulates water movements. So the specific movement of water from nephron to the blood in the presence of antidiuretic hormone is referred to as facultative water reabsorption. So we can write over here the osmoregulation. And would like to write further information. And the third is the water and the molecules with a molecular mass of less than 68,000 from blood to nephron. From blood to nephron. It means the process of filtration takes place over here. So which process is taking place? That is the ultrafiltration, in which water and molecules with a molecular mass less than 68,000 are filtration process. So this process is called as the ultrafiltration. Now the next question is, identify the regions of nephron represented by P, Q, and R. So we are moving towards this question. Identify the regions of the nephron represented. And P, you know, the absorption of the different nutrients was taking place over here. So selective reabsorption was taking place over here. So highly regulated process in the, which is taking place in the proximal convoluted tubule. Proximal So it takes place in the first part after filtration, where the proximal convoluted tubule, the selective reabsorption of these glucose, amino acid, and water. So it takes place over here. Selective reabsorption in the proximal convoluted tubules takes place. And the second is, the facultative water reabsorption or the osmoregulation, in which the water moves from the nephron to the blood in the presence of antidiuretic hormones. So this process occurs in the distal convoluted tubules and collecting ducts. Distal convoluted tubule or and collecting tubules. So the water reabsorption takes place in both parts of this nephron. And the third, in which is the ultrafiltration, ultra filtration of water and the molecules with a molecular mass of less than 68,000 small solutes, move from the blood to the nephron. So the process of ultrafiltration is taking place over here. So this non-selective state of urine formation is driven by the high blood pressure in the glomerulus, and it takes place in the Bowman capsule. This Bowman's capsule, because the ultrafiltration process takes place from glomerulus to the Bowman capsule. And next question is, a student cut a fresh kidney lengthways and placed one half in the freezer. After 24 hours, the student examined the kidney section and tested its firmness with a mounted needle. Sodium chloride concentration affects the freezing point of a solution. So the student drew the diagram figure 6.1 to show an area that had frozen hard and an area that was softer and less frozen. So the hard frozen, which is cortex, and medulla portion, soft and less frozen. So they have labeled them with A and B. So the question is, suggest an explanation for the observations in figure 6.1. So freezing point for these two can be the core, the freezing point depression adding. When we add a solute into a solvent, it lowers the freezing point of the solution. So higher solute concentration also lowers the freezing point. So the kidney structure is designed to create a concentration gradient for efficient water reabsorption. So these can be, I would like to write over here that I will write the proper answer on the given place. So adding a solute to a solvent lowers the freezing point. And lower solute concentration, there will be the higher freezing point. Lower solute concentration will implies implies the higher freezing point. So this is the common rule. So the kidney structure is designed to create a concentration gradient. Concentration gradient for efficient water reabsorption. So I would like to write now the answer. After this, the difference in freezing is due to the medulla. Medulla is B, having a much higher sodium chloride and solute concentration. Then vortex A, which lowers the freezing point of the medulla to a temperature below that of the freezer, preventing it from freezing completely hard. So it was all about question number 6. Please like, share and subscribe my channel. Thanks for watching. For more videos on this paper,